Okay, so in this section, we're going to talk about lecture 4, the microbial growth and the nutrition. So first of all, bacteria growing, we call it a binary fission. Basically is one cell will become 2, then 2 cells become 4, 4 become 8. That is called a binary diffusion. So when you see the picture here, that's DNA attached to the cell membrane. Then they will be enlarged and duplicated. And then there is a wall will be formed. And then the DNA send into the 2 exact daughter cells. And then the cell will be separate. Okay, so that's called a binary fission. 1 become a 2. Now, ideally, 1 bacteria become 2 bacteria. That needs to be exactly the same. But you will see sometimes there is not really exactly the same because there is a mutation happens during the DNA replication. Okay, something I want to mention. Now, the second question is a generation time. How you understand the generation time? Generation time, very easy to understand, is a doubling time. The time required for a population to double in number. Now, some of the bacteria, their generation time, the doubling time is very short. For example, E. coli. E. coli can grow in the broth medium as soon as 18 to 20 minutes. The doubling time is very short. But some of the bacteria, like we mentioned, the tuberculosis, the Mycobacteria tuberculosis, their doubling time is very long. It takes 12 to 24 hours. This is the reason when we do to identify Mycobacteria tuberculosis, we have to use acid-fast stain to do the identification instead of waiting for a couple of days, then we can testing Mycobacteria tuberculosis. When we get a sputum sample from the patient, based on the X-rays, based on the BCG vaccine testing, all serious testing, we need to get a sputum sample, then do the acid-fast stain. If we find acid-fast stain, we will say this patient is presumptive tuberculosis. Then we get a sputum sample, transfer into a reference medium, then transfer to reference CDC reference lab to do the identification. But because Mycobacteria tuberculosis, the doubling time is as long as 24 hours, so we cannot wait that long. When we see the acid-fast stain positive, we have to do the treatment. For example, use antibiotics. Okay, so this is generation time. Now we can calculate the generation time. So how we do the calculation? Look at the formula here. This is NT equals N0. N0 is the initial population multiplied by 2 times N. This is N is the number of generations in time T, which means number of doubling times. And then we change it to the log base, which is log base 10. Now for bacteria cell, we usually change it to log 10 base. The reason is the bacteria cell usually it's a lot. It's more than 1 million, 1 billion sometimes. So if we only show the raw number, we cannot do the statistics analysis and the comparison because the statistics error is too much. So we usually change it to log 10. So when you become a log 10, you can do a simple math here, and the log 10 2 equals 0.301. So when you do the transfer here, you will see this K, the growth rate constant K equals this formula. Now, how you calculation the doubling time? If a population doubles, the NT equals 2N0. So when you replace NT with 2N0 in this formula, then you get something from here. So then you concise it and transfer it Finally, the generation time is reciprocal of the growth rate constant. So G equals 1 divided by K. Okay, so that's the generation time, how we do the calculation. Now for bacterial growth, you need to know it's not linear. Bacterial growth has two phases. So this is a bacterial growth curve. Y-axis is the log number of viable cells. X-axis is the time. So you will see the curve at the beginning is very flat. That's called the lag phase. During the lag phase, basically the bacteria do the preparation, including they set up everything, get accustomed to the new environments, and get ready to do the replication. The second phase, we call it log phase, or we call it exponential phase. This phase is growing dramatically, growing rapidly. Usually the doubling time is happened during that period. Then later on, the bacteria will go into a flat phase, which is called a stationary phase. When you see the flat curve here, which means their death and the growth start to have the balance, because at that time, the nutrition has been exhausted in the environment or in the medium, and then their toxication material from the metabolic process started to accumulate. So at that time, the growth is stopped. Later on, when all the nutrition has been used, and in the median, they are all toxication material. For example, like H2O2, O3, like all these toxication material accumulated, then they start to kill bacteria. So then the number is dramatically decreased, and we call it death phase. Okay, so always remember the bacteria grow including lag phase, log phase, stationary phase, and death phase. Now, when we say that we are curing bacteria, or we say bactericidal effects, which means bacteria stopped at the lag phase, doesn't even reach the log phase period. But when we say we inhibit bacterial growth, that means the lag phase time will be longer, but does not mean it's not gonna grow. It will grow later on. Just delay the growth, which means the lag phase time will be intended. So this is for the microbial cell growth. Now, something else I want to also mention is that we know we can grow bacteria in the medium, in the liquid medium, solid medium, which means the bacteria is culturable. But you also need to know, in the real life, there is some of the bacteria is viable, which means they could be cause problem. They are still alive but dormant. They can grow, they can be pathogenic, but they are not culturable. So we call them viable but non-culturable. Second, the bacteria entering into the death phase is not going to die immediately. They will have a programmed to be died. This is the same thing in the human cells. We have a programming die process. Once the programming die process has a problem in the human being, usually we're going to end to the cancer. And for the bacteria, we call it commit suicide. Quotation mark, commit suicide, because it's genetically programmed to die. Okay, so that's for the bacteria, the death phase. Now, what are the factors will impact or affect microorganism growth? Of course, there's a lots of factors, but basically what we're talking about here is pH, temperature, water activity, and oxygen concentration. So let's talk about these one by one. First of all, pH. What is a pH? This equals minus log 10, the concentration of hydrogen ions. Now we know that neutral pH is pH at 7. This is the optimal growth pH for most of the microorganisms. Usually the pH below 4.0, which means in a very acid environment, Now, most of the pathogen with the growth will be inhibited. When the pH below 5.0, the yeast and the molds can still survive, can still grow, but bacteria will not be grow probably or grow slowly. Now, based on the pH, the requirement for the growth condition of the pH, we can classify bacteria into 3 categories. The first one is acidophile. Most of them are fungi. pH 0, their optimal pH is 0 to 5.5. Second, we call it neutrophiles. This refers to most bacteria. Their pH is between 5.5 to 8.0. The last one is for some of the bacteria, they do like in alkaline environments, which means a higher concentration of hydrogen peroxide. So that's alkaline phials, which means the pH, optimal pH is 8.0 to 11.5. The examples are Bacillus, Micrococcus, and Pseudomonas. Now be very careful. There is one thing happened in the meat industry, happened in the food industry a lot, which is called acid tolerance. So we all know that when we are lower pH in an acid environment, let's say pH at 4, the bacteria will stop to grow, or growth will be inhibited. But bacteria also can survive because there are some mechanisms. For example, they will make their plasma membrane permeable for protons. By doing that, they exchange with potassium. Second, once they pump the proton out of the cell, they will be synthesized acid and heat shock proteins. Therefore, later on, they become acid-resistant bacteria. This is a huge problem in the meat industry. So we know when we sanitize beef carcasses, we're using lactic acid bacteria a lot. So when we spray on the surface, most of the bacteria will die. But some of the bacteria will survive, then become acid-resistant. Once they become acid-resistant, they have activated heat shock proteins. What that means, which later on that carcass become a beef jerky or beef steaks. Then they're going through cooking process. Usually the normal cooking time may not be kill all of them because the acid stress adaptive cells generate cross-protection impacts by heat shock proteins. Okay, so that's is a big problem in the meat industry. So usually in the industry try to solve this problem, they not only use one type of the antimicrobials. For example, they use lactic acid, they try to use acetic acid, or they try to use hydrogen peroxide or peroxyacetic acid to do rotation, or at least 2 or 3 different types of the antimicrobials. They do the rotation. But they also could cause a problem. Later on, the bacteria will become a superbug, which means they are resistant to multiple antimicrobials. So just to let you know that. Temperature. Okay, bacteria cannot regulate their internal temperature, but they do have an optimal temperature. They can function optimally. The reason is that the temperature is highly related to the enzyme function. So you know that when you have a fever, when you have a 39 degrees Celsius, or you more than 100.4 degrees Celsius, or degree Fahrenheit, you have a fever, you're not gonna feel comfortable to eat because the enzyme is unfunctional or dysfunction with higher temperature. The reason is the enzyme's optimal temperature usually around 37 degrees Celsius, okay? So for bacteria, their growth temperatures, we have a minimum, optimal, and a maximum. Based on their optimal or the minimal temperature for the growing bacteria, we can categorize them into 4, which is psychrophiles, psychrotrophs, mesophiles, thermophiles, and hyperthermophiles. Now, psychrophiles, usually it's in the ocean and in the marine area, like Arctic area. Okay, those marine ocean area, in the deep sea, they are very cold. So the bacteria survive there. They become psychrophiles. Now, cyclotrophs, this group of bacteria, they can be growing in the refrigerated temperature, like Listeria monocytogenes, like Pseudomonas. This causes a big problem for the food safety because people always think about you put food into the refrigerator is safe, but not really. When you have Listeria monocytogenes on the ham, on the turkey breast, when you vacuum package, even you do vacuum package, that you put in the refrigerator, they can still grow. And they can grow very fast. Within 3 months, they can grow from 0 to 9 log. So that means those bacteria can accustom to the very low temperature. So that's belonging to the cyclotrophs. Now, the third group, which is most of our bacteria or most of our foodborne pathogens, is belonging to called mesophiles. The optimal temperature growing is 35 degrees Celsius. Now, the next one is thermophiles. One group of bacteria belonging to that is bacillus. Bacillus can survive very high temperature, as high as 60 to 70 degrees Celsius. So bacillus usually to do the validation study of thermal inactivation in the food products, or they also can use it for to testing some of the cooking process. The last group is the hyperthermophiles. Hyperthermophiles, most of them is environmental bacteria. They can survive in very high temperature, even 100 degrees Celsius. Which means you using boiling water, you will not kill them. So those bacteria usually is a good example is in Yellowstone, in those hot spring, you have those hyperthermophiles. We do have another example for the hyperthermophiles is geobacillus. So geobacillus can survive more than 100 degrees Celsius. So we were usually using geobacillus to test the impact of autoclave, because the autoclave machine needs to be reach temperature 121 degrees Celsius. If the autoclave can kill geobacillus, which means it's functional. If not, if the temperature cannot reach 121, for example, if it's unfunctional, only reach 105 degrees Celsius, then the bacillus will still survive. The geobacillus will still survive. Okay, so hyperthermophiles, most of them is environmental hot spring bacteria, but we do have an example in the lab, which is geobacillus used for validation the function of autoclave. Now, this is a temperature range for the microbial growth. So you see the cyclophiles, 0 to 20 degrees Celsius, cyclotrophs, 0 to 35 degrees Celsius, mesophiles, 20 to 45 degrees Celsius. So like I just mentioned, the mesophiles belonging to most of the bacteria. So like E. coli, Salmonella, they won't grow in the refrigerator temperature because their range is 20 to 45 degrees Celsius. That means they can grow very well in the room temperature because room temperature is about 25 degrees Celsius. Thermophiles, the range is 55 to 85 degrees Celsius. And the hyperthermophiles can be as high as 113 degrees Celsius. So that is a temperature range for the microbial growth. Now, the next concept is talk about water activity. Now, what is water activity? Remember, we talk about the water content or what amount of food products. That is talking about moisture. Now, water activity is not, it is related to the moisture, but not equals moisture. Water activity, which means the index of available water. You can easily understand is how much water amount the bacteria can use. That's called available water. It is calculated by the pressure of the solution divided by the pressure of the pure water. Now, what is the vapor pressure? That is the rate of escape of molecules, which means how fast the molecule can be evaporated or escape from the surface. Now, usually you need to know the pure water is 1.000. No water is 000. when you add sugar or you add salt ingredients into the water, then the water activity will be decreased, because any of the salts or the sugars adding into the solution, then the free water will be reduced and the escape rate will be decreased. Okay, so be careful about that. Now, how they calculated the relative humidity, which is calculated by 100 multiplied by the water activity. Now, just to give you some example, when we talk about hot dogs or turkey breast, those foods are very easy to get contaminated with pathogen, like Listeria monocytogenes, because those food, the water activity is very high, is as high as 0.992. But like beef jerky is the dry products, is not easily to be contaminated with pathogen, because the beef jerky, the water activity is only like 0.9 or 0.85, same as some sausage and salami. Now, when you see the powder, the milk powder, those usually only the sporation bacteria will grow there. Lots of the bacteria won't be grow because the water activity is as low as like 0.3 to 0.4. But be careful, even at the lower water activity, the yeast and the molds can still grow there. Okay, this picture here, which showed, this is actually we are mentioning is called osmotic unbalanced. So this is a normal bacterial cell and the cell membrane. Once you put in a high concentration of dissolved components, or let's say we put in a salt solution, it will become shrink. You know that. That's called the plasmolysis, because unbalanced of the osmosis inside outside of the cell wall, then it becomes shrink. Okay. In other words, if inside of the cell has a higher concentration, the outside is like pure water, like a distilled water, it may become burst. That's why you put the bacteria in a distilled water, you will see it can kill a little bit of bacteria, because some bacteria will be burst. Okay, so that's water activity. Now, another thing, since we talk about the water activity, we're gonna talk about the sodium chloride on the bacterial growth. Now you need to know sodium chloride, salt, which is a natural antimicrobials. In the salt solution, lots of the bacteria can be died. So if they died quickly, we add 1 mole sodium chloride, it will be died rapidly. We call it non-halophiles. Most of the bacteria is moderate halophiles. But we do have a bacteria which is halophiles. For example, Staphylococcus aureus. They can grow optimally at 0.2 mole with salt. Now, lots of the bacteria in your mouth, in the teeth, it's also sodium chloride, resistant to the sodium chloride. We can easily understand the Staphylococcus aureus is halophiles. Why? The most predominant places for Staphylococcus aureus to survive is on the skin surface. But remember, your skin surface will be extracted with sodium chloride all the time. When you go to run 100 meters, you are sweating. Is that right? Then the sweat comes out of the sodium chloride. So on the skin surface, the most dominant bacteria actually is Staphylococcus aureus or Staphylococcus epidermis. All those Staphylococcus actually resistant to the sodium chloride. So they call halophiles. Now we also gonna see some of the extreme halophiles. They can survive in the temperature, in the sodium chloride concentration more than 2 mole. Those also are environmental microorganisms. And where you can find it, the good example is all the microorganisms in Great Salt Lake, in Salt Lake City, Utah. So when you get a chance to go Yellowstone, okay, you're parking at Salt Lake City Airport, you drive 4 hours, you go to the hot spring at Yellowstone, you will see lots of the hyperthermophiles. When you go back to the Great Salt Lake City, in the Great Salt Lake, you can see lots of the extreme halophiles. So that area is very good for the environmental microbiologist to study all the environmental bacteria. Now, another tricky thing is that all those environmental bacteria or microorganisms, when they survive in those very extreme conditions, if you knock out of their genes, you purify those genes, usually you will get some of the dramatic stuff, because that's a way the bacteria can survive in those very harsh and extreme environmental conditions. Okay, this is the slides when we talk about oxygen. So bacteria basically have three categories. Number one is the bacteria must use oxygen to grow. They will die without oxygen. So we call it obligate aerobics. Obligate aerobic bacteria. There is some of the bacteria is strict anaerobic, which means they cannot survive in the presence of oxygen. Once you have oxygen, even a little bit amount, they will be died. Now, most of the bacteria, we talk about the foodborne pathogen, E. coli, Salmonella, Listeria, all those, we call it facultative anaerobic, which means they can grow in both in the presence, absence of oxygen, but grow better with oxygen. Now, why they will have so many categories? They will have this situation. This is something you need to know. When the bacteria grow, they are going through metabolic pathway. The metabolic pathway coupled with electron transfer. The electron will transfer from NADH, go to FAD, go to COQ, coenzyme Q, then go to cytochrome C, cytochrome AA3, and oxygen finally. During that process, they will generate a toxication material, such as H2O2 or O3. So we need enzyme to hydrolyze them. Some of the bacteria have SOD, disulfide oxidase, and the catalase, disulfide dismutase, and the catalase. They can be hydrolyzed those toxication material, so they can grow there. But some of the bacteria, they cannot survive in the presence of oxygen. The reason is they do not have neither of them. They have neither of those two enzymes, which means if they expose to the oxygen, they actually, which equals they expose to the toxication material, so they will be died. The good example is Chloride. Remember, we talk about the endospore. They will generate endospore. They'll talk about the Chloride perfringens, difficile, all those Chloride, they are actually cannot survive in the presence of oxygen because there's no SOD and the catalyst. Now, either you have one of them, you have another category comes out, which is allotolerant anaerobes. They grow equally well with or without oxygen because they have SOD, no catalyst. Now, another group called the microallophiles, they require to grow with very little amount of oxygen, 2 to 10%, damaged once oxygen is more than 20%, because they have SOD, but very low level catalysts. A good example for microallophiles is Campylobacter. The Campylobacter jejuni, Campylobacter coli, that is a major problem, major pathogen for the poultry industry, of the poultry meat. Okay, so this is, we talk about the requirements for the oxygen. So basically there's aerobus, anaerobe, and the facultative. Why they will have those three basic categories depends on whether bacteria has SOD and the catalyst. The reason is SOD and the catalyst will be hydrolyzed the toxication material during the metabolic pathway. If you don't have any of them, then bacteria cannot survive because the oxygen is a toxication for them. So they have to be strict anaerobes. Okay, so that's a basic story. Now, but depends on the amount of their SOD and the catalyst, they further can categorize, become aerobic tolerant and the microallophiles. So that's a conclusion of these slides. Now, how we grow anaerobic bacteria. We can use two methods. First one is a very expensive. This is called the anaerobic chamber. This chamber is as cheap as $25,000 to $100,000. There is a pump there and there is a gas generator there. They will be extruded all the oxygen out. So you put your hands in those two arms, they go inside, then you do all the operation inside. This is for the large lab to do the anaerobic bacteria. Most time in our lab, we are using this jar. This jar costs usually $400. So the story is that we put bacteria plates there. We add a gas pack. Then we add the water there. Once we add the water, it will generate hydrogen and carbon dioxide. And the hydrogen will be reacted with oxygen. Because oxygen reacted with hydrogen becomes a water. If you want to have this reaction happen quickly, then underneath the cap, you need a palladium pellets, because this will let the reaction happen very soon and efficiently. Then how do you know all the oxygen are gone? We usually put an oxygen indicator. Here we put a methylene blue. The methylene blue will become colorless in the absence of oxygen. So no oxygen, it will become colorless. Once you see, you put a gas pack, you put a water, you seal it. After a couple of hours, you see the methylene blue strip become colorless. The blue is gone, which means all the oxygen are gone. Okay, so that used to do an indicator of the oxygen. We also can use resazurin. That's another indicator. Resazurin at the beginning is become a pink. Then later on, if it is colorless, then which means oxygen are gone. So that's a strict anaerobic bacteria and how we do the cultivation for that. Now, another thing, most of the time happened in the industry, in the food industry, is for a biofilm. You can understand on the surface, there is a poor nutrition there, but bacteria can grow. They not only grow there, they will be grab the neighborhood and become a heavy matrix. And because of the heavy matrix, then they will become resistant to the antimicrobials, to the sanitizers. So the story is there on the surface, even if it's poor nutrition, you more or less have some moisture. As long as you have a moisture, the bacteria will be attached to them. Okay, at the beginning, they based on the van der Waals force, which is very loose attached. But later on, they will be separate. Some of them attached, some of them released back or dispersion to the air. Then once the bacteria stay there, they will do a communication with each other called cell-to-cell signaling. We have a terminology for that, which is called chlorine sensing. And once they're doing the chlorine sensing, they will be formed a very heavy matrix that is composed by polysaccharides, something like a very heavy peptide cell wall. Then they become a matrix, so they become very, very heavy. And later on, some of them will be detached, some of them will be stay there. That could be longer than 1 week, 1 month, or even a couple of years. Now these matrix, the major problem, these matrix, because of they do the chlorine sensing, they stack with each other. So you need to use a higher amount of the antimicrobials to do that, because they're resistant to the antimicrobials and the sanitizers. The research showed that compared to the bacteria growing in the medium, we call it the vegetative cells, the bacteria grow in the surface, on the surface. Once it's formed the biofilm, it need 1,000 times higher concentration of the chlorine compared to the vegetative cells to be killed or the bacteria. So that's always a big problem for the industry. As long as they have a residual bacteria on the food contact surfaces, then the second time to do the sanitizing, the bacteria will become resistant to those sanitizers. And the more become resistant, the more and more heavy to do that. So more important to control the biofilm is you have to do a physical removing with chemical antimicrobials based on the daily sanitizing procedures. That is referred to sanitation operation procedures, which is called SSOP. That is a prerequisite program for the proactive food safety plan, which is called hazard analysis critical control point, which is called HACCP. And we're going to talk about that later on, okay? So from here, we will end. Then next module, we'll talk about cultural media, okay?